Hi there, I'm Alex DiGiorgio, co-founder and CEO of Sable Bio, and today I'm going to give you a brief introduction to parts of Sable's Target Intelligence platform. We're aiming to make target-based safety more quantified, more systematic, and more actionable. The scientists we speak to say compiling target safety reports takes much too long. They worry they're missing important information, and they struggle to combine and then prioritize relevant safety data. And for today, we're going to look at an example of a well-known target on our platform, a GLP-1R agonist that came to fame as mechanism of action of drugs like Ozempic. Let's have a look at Sable's Target Intelligence report for GLP-1R activation. The first thing you'll notice is our high-level overview of potential risk areas. On the left, there's a radar plot to flag risk levels across key organ systems, focused on organs most implicated in trial safety failures. On the right, you can see a summary bringing together a range of evidence types that give you a picture of what we'll be looking at in the rest of the report. And below, the target intrinsic risks, each of which is correlated with risk of clinical safety failure. In GLP-1R's case, we can see it has enhanced expression in specific tissues, interacts with relatively few proteins in this specific protein interaction network, is highly but not very highly genetically constrained, and is not a tumour suppressor or oncogene. On their own, these aren't enough to make a clear safety assessment, but you'll find these often correlate with the degree of potential risk elsewhere on the report, and so can give a good first impression of the sort of target that you're looking at. Next up, the key risk organs associated with GLP-1R agonism. We can see the gastrointestinal system, the central nervous system, and the cardiovascular system, which are key areas involved in this drug target, reflecting its widespread involvement in regulating metabolism, appetite, and glucose homeostasis. Going into GI, we can explore groups of potential risk phenotypes. Within this view, you can see individual phenotypes considered to be potential risks given your chosen direction of modulation, which in our case is an agonist, an activation. You'd see different risks if you're choosing to inhibit GLP-1R, which would be considerably less safe. On the right, we have a brief reference summary of key supporting evidence. For today, we'll look at nausea, given it's quite a common adverse event seen in trials. This is somewhere the Sable platform comes into its own. You can see an interactive view of the evidence, allowing you to explore the underlying data yourself. Our platform's combining a range of data types, from expression, genetics, and scientific literature, to mouse knockouts and clinical evidence. Here for nausea, we can see a summary of the data that supports this potential risk. Below, we can see the individual types of supporting evidence, in this case coming from FDA post-marketing reports and the scientific literature. We use advanced methods to assess direction of effects, indicating that this phenotype may increase when activating GLP-1R. Most importantly, clicking through, I can see specific paragraphs of text that support that assessment and link out to individual papers. So that was a whistle-stop tour of parts of Sable's Target Intelligence platform. We're continuously expanding our platform's capabilities, so I'll be posting more of these videos for deep dives and new features. Our key value proposition is to help you make higher quality safety decisions, while making the whole process much easier at the same time. If you want to learn more, visit our website and reach out to our team, and we'd love to schedule a call to explore in greater depth. Thanks for watching.